Hi, it's Mr. Young, and in this video podcast, we go through looking at two big molecules that we have in our bodies, carbohydrates and lipids. You're preparing for your big 26.2 miler this weekend, your first marathon, and Friday nights come and you've been determined to do some carbo loading. And so you order up your favorite dish of pasta because you've heard that carbo loading is what you need to help you get past that mid-marathon wall that you may be hitting. Well, how? Why? What's this pasta going to do for you tomorrow when you're eating it today? A good understanding of the basics of biochemistry will help you in figuring out why you're doing this. And it all starts with taking some pasta and putting it in your mouth. And as you do so, you're essentially taking a larger molecule shown here that we call starch. And what we see is that this starch molecule, which actually goes much further than this, it's elongated as a big chain. The starch molecule is made up of a bunch of repeating units shown here and here and here. They just keep repeating and repeating and repeating. Incidentally, each one of these units is a glucose subunit. And so glucose molecules are put together in a big long chain to form starch. But once you take that starch molecule that you got in your pasta noodle and put it in your mouth, the digestion actually begins. Your body starts to break it down. And with the help of an enzyme called amylase that's produced there in your salivary glands, we end up taking our starch molecule and we break it up in smaller chunks. Our chain gets broken down into groups of 2. And then each group of 2 gets broken down into groups of 1. And eventually you have, roaming through your blood, little tiny glucose molecules that are neighbored by white blood cells and red blood cells. And that happened after that noodle was swallowed and you broke it down. And then later on in your intestines, we see absorption of that glucose into your bloodstream. Well, there in the bloodstream, the blood will take those glucose molecules and will ship it to wherever it needs to go. Like, well, your heart as your heart beats, and your muscles as your muscles contract as you chew your pasta. You move your masseter muscle. Because that glucose molecule will be needed in the process of cell respiration to be broken down to produce ATP, a currency that the body recognizes and can use to help power your muscles, among other things. Well, that's today. My race isn't until tomorrow. Why am I eating this now, you may be wondering. Well, as it turns out, your body can't use all that glucose that is currently raging through your blood as you're eating your pasta and use it to make ATP right at the moment. Rather, your body is going to take some of this glucose out of the bloodstream and store it in a location that can later be tapped into. And that storage molecule is shown here. This storage molecule is none other than glycogen. Glyco referring to sugar or sweet. It's a big sweet sugar molecule that, unlike starch, is now more branched, as you can see. And this molecule, glycogen, we can store here in the liver, or we can store down here in the muscles. And what are we going to store it for? Well, we're going to store it for that time in your marathon when your glucose in your bloodstream has now been reduced because of the fact that you've been burning it as you've been powering your muscles, using the glucose to be broken down later into carbon dioxide and to produce ATP out of it to keep contracting your muscles as you race on and on and on. And when your glucose in your bloodstream gets low, the body can say, hey, I've got some of that glucose packed into my liver, packed into my muscles, and now I can start breaking it down into smaller subunits of glucose monomers, take it and put it in the blood, and then once it's in the blood, get it to those muscles so they can keep working and working and working. And so that, ladies and gentlemen, is why you're carbo loading. And so we have this whole concept of taking a larger molecule and breaking it down with the help of enzymes into smaller subunits shown down below. And this is a general process that you're going to see repeated time and again in working with macromolecules in the body. The technical term we describe is what we call hydrolysis, where we're going to use water, shown here, to help to break or split apart or lyse our macromolecule into smaller subunits. Conversely, we can take our small subunits and build larger subunits in a process of what we refer to as dehydration synthesis. In other words, we are going to synthesize a larger molecule out of smaller molecules. But in the process, with the help of enzymes, we end up losing a water molecule, or we get dehydrated. Hydro for water and the D referring to we're losing it. And so these two examples of macromolecule creation, as you can see here, or digestion, as you saw in the previous slide, are going to be pretty common through our various macromolecules we'll be looking at today. Whether we're taking monomers to the polymers, mono meaning one, and poly meaning many of them stuck together, or going in the reverse, taking a polymer to form monomers. We'll be using enzymes in the process. Well, the first macromolecule we want to look at is carbohydrate. And a carbohydrate is none other than a carbon that's right, hydrated, H2O. And that actually is a great simplified formula or empirical formula that we'll see for a lot of our carbohydrates. Whether you're looking at monosaccharides, disaccharides, or polysaccharides, they'll all share that similar empirical formula. For every single carbon you've got 2 hydrogens, and for every carbon 1 oxygen in a 1-to-1 ratio. Well, we'll classify our carbohydrates based on really size. Whether we have a mono or a single sugar unit, that saccharose root is referring to sugar or sweet. Whether we have di or 2 sugar units, again saccharose, or whether we have many or polysaccharides, simply a size classification that we'll look at for our carbohydrates. In the first case, you see how we have single ring structures, single as in one, like a monosaccharide. In these cases, you have single ring structures that actually can exist in a linear form, where they are just a chain of carbons, but they can, in solution, form ring structures. In this case, what you're looking at is a molecule called glucose. The very molecule we looked at before, your body will end up reducing starch into. And in this glucose molecules, you can count your carbon, your 1, 2, 3, 4, 5 carbons down below in the ring, and then an extra carbon or a 6 carbon down here, or C6, H12, you can count your hydrogens, and O6. How about it? There you go. Well, over here in the other simple monosaccharide or simple sugar, likewise you can count your carbons. Here in this organic chemistry kind of notation, we will identify every corner that you see as a carbon. So there's a carbon, there's a carbon, there's a carbon, there's a carbon, there's our 6th carbon. We still have 6 carbons, C6. Let's count our hydrogens, where now we have 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, H12. Hey, if our initial formula where we saw we have hydrated carbons holds true, how many oxygens should we have? That's right, we should have 6 oxygens in a 1-to-1 ratio with carbons. And do we, in fact? There's 1, 2, 3, 4, 5, 6. How about it? Different molecule, though, isn't it? It looks different, but it has the same chemical formula. This is what we're looking at when we look at a molecule of fructose. And we're looking at none other than isomers. That's exactly right. Isomers, or molecules with the same chemical formula but different structural formulas. Glucose, as we see, is going to be essential for our production of ATP, and so it's a key energy storing compound, energy within the carbon-carbon bonds. And yet we see we can have a higher degree of complexity when we add these monomers together to build, in this case, a disaccharide. So what do we have? We have a glucose monomer shown here, glucose, and then a galactose molecule right next to it. And if you were to count their carbons, and if you were to count their hydrogens and oxygens, we still have the same chemical formula of C6H12O6 for both of them. C6H12O6. And so if you were to combine these, what do we have? We formed a sugar lactose. Have you seen the pattern here that repeats? Ose, galactose, glucose, fructose, lactose. It goes on and on and on. This is milk sugar. And you would expect if you were to add up these 2 monosaccharides to form a di, or a 1 2-ring sugar, what would be the chemical formula? Yeah, you'd anticipate, hey, we'd have C12, wouldn't you? 6 plus 6 is 12. H24 and O12. Expected, yes. Accurate, no. You see, what we find happening is, if you were to count your carbons, we do see 12 carbons, but our hydrogens are off. In fact, rather than 24, we have 22 hydrogens. And we lose a single oxygen. We only have 11. Bummer. We lost 2 hydrogens, and we lost a single oxygen. Hey, that looks familiar. We gave water out in the process of synthesizing a larger molecule. Once again, a great example of what we call dehydration. Notice we lost water, we got dehydrated, but it was a synthesis reaction because we synthesized or we built a larger molecule. Dehydration synthesis. Great example of how disaccharides can form, in this case lactose. Well, we can even build bigger sugars, in these cases polysaccharides. And polysaccharides here can serve a function in energy storage. That's what you see here in the case of starch in plants and glycogen in animals. However, they also have a structural component to them, and that's what you see here in the form of cellulose or chitin. Structural polysaccharides used for cell walls in plants, used in this case for the exoskeleton of things like this stonefly that you can see. Sometimes you'll flip over rocks in the winter, or rather in the summertime as you're swimming on your favorite river, and see these apparent shells or casings of aquatic invertebrates. And this is an example of the casing that's made up of a chitin, or a polysaccharide that also makes up the cell walls of fungi. Our various polysaccharides, as we mentioned before, have structures that indicate monomer units that are bonded to one another. These little glycosidic linkages that you link them together. Whether it's starch as a food storage or energy storage molecule in plants, or glycogen, the energy storage molecule in animals, they show repeating patterns of their monomer units that were assembled together by way of dehydration synthesis. In addition, we see variation in those chains. In some cases, we have highly branched molecules, as is glycogen as opposed to the starch. In other cases, we have repeating patterns of linked monomers of glucose that here have some intermolecular attraction here, showing the hydrogen bonding of cellulose. Regardless, great energy storing molecule, carbohydrate for short-term energy use, or in the case of the structural components like cellulose, great structural pieces that help to facilitate stronger cell walls, as is cellulose in plants conferring, and the chitin conferring in fungal cell walls. Well, another great molecule or large molecule we have in the body is shown here down below, where you see a picture of a bunch of fat globules. These are called adipocytes, or cells literally that are fatty in nature, adipo. And so this is adipose tissue, in our case shown to be below the skin or subcutaneous. It is that which is comprising the hypodermal tissue of our skin. And we see that this stuff is not only found in our adult skin hypodermally, but also it's located in different areas of infants. And as you see here, you are seeing what we call brown fat, and it's a special kind of fat that takes on a coloration characteristic of the abundance of mitochondria within. And whereas in the adult animal we see a high degree of adipose tissue that is subcutaneous, this helps to serve a role in thermal regulation, regulation of body heat. Little kids, however, may not have as much of that, nor are they able to shiver perhaps as much as we do and generate as much body heat through that. And so they have brown fat, which is actually there as a thermogenic molecule, helpful to regulating body heat in our little kids. And we see it around the torso, around the midline there, around the kidneys, and on the back as well. But beyond the role of regulating body heat, of perhaps cushioning, as you can see our fat will do around organs, for example, we also see that fat and lipids provide a great source of energy. In fact, a misconception you may be that fats actually provide more energy per gram than carbohydrates. Yes, carbohydrates are a quick fuel source for us, but long-term energy source are what our fats will provide. Just how fats provide great energy for us can be found in the molecule of a triglyceride. Triglycerides are actually the fat stored in those adipose cells or adipocytes that we previously saw. And triglycerides have these 3 or tri fatty acids that stick off the glycerol molecule. And the glycerol molecule, if we look at it, looks nothing more than really a sugar unit, because you've got carbon shown here, carbon shown in all these big black balls, You've got hydrogen shown in the little black balls, and then you've got oxygens shown here. Boy, if you're just to look at the chemical formula, you might be thinking, hey, we've got C's, H's, we've got O's. That looks a lot like a carbohydrate, but the difference here is in the proportion of oxygens to carbons, for example, where our ratios are quite a bit off, aren't they? Many fewer oxygens that we have here, and tons of carbon, hydrogen, or hydrocarbon units that make these long chains. And that's what we see here when we look at the overall structure of our fatty acids connected to our glycerol backbone. That we see carbons bonded to one another and long, long, long chains of hydrocarbons that continue. In fact, it's these hydrocarbon chains that confer insolubility to this molecule. Insoluble in water because they lack the polar nature, being non-polar, that you saw in other compounds, even other sugar compounds, making these not able to dissolve in water. They don't like water. These are fats, after all. And among the different kinds of fats that we see, we have saturated fats, you have unsaturated fats, but what in the world is the difference about them? Here we've shown a single fatty acid group right here. And what I want you to pay close attention to is how this fatty acid group off the saturated fatty acid compares to the fatty acid group off of an unsaturated fatty acid. What's the difference in structure? Do you see it? Good work. At this point you've seen that, hey, we've got double bonds here in the unsaturated fats, but in the saturated fats, there are carbon-to-carbon linkages that are all single bonded. And what that means is that every carbon here is saturated with as many hydrogens as it can potentially have. That's where the saturation comes from. Every carbon here, if it's bonded to other carbons, has 2 hydrogens, whereas over here, because carbon wants to only have 8 electrons that it's sharing, we see that this carbon only has a single hydrogen. It is not saturated, or it is unsaturated with hydrogens. As a result, we see some difference in the properties as well. Whereas the saturated fats are stackable, notice how they are very much linear. And if this were to be the shape of a little toy piece that you were using to build a big tall tower, you could build a lot of tall towers with it. But if this were to be the shape of your piece you're using to build a tall tower, due to the kinking of these saturated or unsaturated fatty acids, it would fall over. That's exactly what we see in the body as well. Being that these unsaturated fats are liquids at room temperature versus solids at room temperature, the solids are much more stackable because of the single bondedness of the saturated fatty acids. Stackable, single bonded, saturated fatty acids. Little thing to remember, those saturated fatty acids versus your unsaturated fatty acids. Both types of triglycerides, which are the body's most concentrated and most abundant form of energy. Not carbohydrates. Yes, we have carbohydrates, a great form of quick energy, but the most concentrated form of energy, triglycerides. Well, where else do we see lipids in the body? You don't have to look any further than the membrane of every cell in the body, which is comprised of phospholipids. And here, as you look at the phospholipid bilayer, we can zoom into a single phospholipid, which had a head and 2 tails, and see how there's variation in the properties as a result of the variation in the structure. Well, where's our hydrocarbon chain? Where's our lipid chain? Those are shown right here, where again, every little corner is a carbon, and we have hydrogens that will come off of all those carbons to satisfy the octet that you learned about in basic chem. But our hydrocarbon chains that come off here as fatty acid tails are the lipid component of a phospholipid. And the phosphate component of the phospho part is shown right here. And what you can see with the negative charge here, we are looking at a unit that is sharing really 2 basic properties. One property over here that is hydrophilic, meaning it is lover of water, and one down here which shows hydrophobic tendencies, meaning it fears, like a phobia, water. And so one molecule having 2 different tendencies, an amphoteric molecule we may call it in chemistry. But this is a molecule that we see comprising every cell in the body, phospholipids, a great example of a lipid in the body. Other forms of lipids that we see in the body are shown here, and this is the unit of cholesterol, which is characterized by the classic steroid 4-ring structure with a variety of different functional groups coming off it. In this case, cholesterol has this OH or hydroxyl unit coming off of this carbon, and then a variety of carbon-hydrogen or hydrocarbon linkages that go beyond there. And cholesterol, while you may hear it, we want lower cholesterol, we certainly do. At the same time, there's a needed amount of cholesterol that can be found within the phospholipid bilayer embedded, helping to influence the flexibility of my phospholipid bilayer. And so this is one great example of a steroid molecule. Other steroid molecules in the body we see are found as sex hormones. And once again, as a steroid, it has the 4-ring structure shown here in all of these different sex hormones. But interestingly, you change very few functional groups and you change the molecule entirely. Notice down below, one's testosterone, androgen, a male sex hormone, the other estradiol, progesterone. 2 critical female hormones that are involved in regulating, among other things, the uterine cycle. And so all of these go to show how in carbohydrates and lipids we see the theme of structure influencing function. One similarity that you see among them, besides that theme, they both can serve a role in storing energy for the body. Carbohydrates, quick fuel source of energy, and lipids, a much longer term storehouse of energy. In fact, to the degree that later on you may learn about lipid synthesis and lipid metabolism, as lipids can be formed from carbohydrates, excess carbohydrates, and lipids can be metabolized to form carbohydrates and to help be used to break down and provide fuels later. Stay tuned later for a look at proteins and nucleic acids. All right.